Greetings, everyone. Welcome. We are starting today the first virtual session of the Xylella Fastidiosa workshop, From Diagnosis to Management. This workshop was organized in partnership between Escola Superior de Agricultura Luiz de Queiroz, ESALC, from University of São Paulo, through the Laboratory of Insect Vectors of Plant Pathogens, and also by Centro de Citricultura Sylvio Moreira from Instituto Agronômico through its Laboratory of Biotechnology. This workshop has the important support of Rede Ibero-Americana para a Vigilância de Xylella Fastidiosa, which is supported by CITED. It also has the support of Department of Entomology and Acarology from University of São Paulo, the graduate program in entomology from University of São Paulo, and also the Division of Communication from University of São Paulo. The goal of this workshop is to provide an updated overview on the plant pathogenic bacterium Xylella fastidiosa, its importance, classification, transmission, epidemiological aspects, management, and certification. It also has the goal of providing, through the practical session, a training on research on vectors and also on the pathogen. The workshop was especially designed for researchers, students, but also professionals working in the area, in companies and government, and also for growers. We have approximately 200 people registered for the virtual sessions, mostly from Latin America and Asia, but also from Europe, United States, and Africa, representing universities, research institutes, companies, and governments. The program of this workshop has an online component with 5 virtual sessions that will happen on Thursdays from 11 a.m. to 1 p.m. São Paulo time in Brazil, with the first session starting today. We will also have practical sessions for a limited number of people that will happen on September 5 to September 7 in São Paulo, Brazil, in the campus of University of São Paulo at ESALC, USP, and also at Centro de Citricultura Sylvio Moreira. For more detailed information on the program, please check the workshop website, which will be listed in the chat of this YouTube transmission. ESALC is very proud of organizing this important workshop because we actively conduct research, teaching, and extension on plant protection. And Xylella fastidiosa is one of the most important pathogens affecting crops such as citrus, plum, and olives in Brazil. ESALC is a century-old institution established in 1901 by Luiz Vicente de Souza Queiroz. It's now part of the University of São Paulo and has formed more than 16,000 bachelors in agricultural, biological, environmental, and social applied sciences, and its 120 years of existence. It also formed more than 10,000 professionals and masters with master and doctorate degrees in 17 academic subjects. It has 12 academic departments with 200 faculty and around 3,300 students. So we are very proud of organizing this workshop, which is part of our mission in training people, especially in plant protection. Now, I would like to invite 2 persons representing the 2 other organizing institutions who will speak on behalf of them. So I'll start inviting Dr. Félix Moran from Valencian Institute of Agricultural Research, IVIA, Spain. And I'm also inviting Dr. Dirceu de Matos Jr., which is the director of the Center of Citriculture, Sylvio Moreira. Welcome.
Hello. Thank you, Yao.
Obrigado.
Obrigado.
Hello,
Thank
Felix.
You.
How are you?
So, fine. I don't know if I can share my slide now or
Sure.
yes. Perfect. Thank you. Thank you, Yao, for the introduction. Well, thanks to all the organizer for the invitation of the opening of Xylella fastidiosa workshop from the diagnosis to the management. First, I'm going to introduce myself. My name is Félix Morón, of course, research from the Instituto Valenciano de Investigaciones Agrarias, or IVIA. And today I'm here to talk briefly about the objective and results of the CTA Versified network. So let's go to start. To start, I would like to comment the reason why this network was created. This motif has a first and last name, and is Xylella fastidiosa, of course. It's a plant pathogenic bacterium which is able to infect a wide range of hosts, causing the devastating disease that are a problem for the agricultural sector. This problematic bacteria, in addition to causing important symptoms in plant, is able to be transmitted by insect vectors that feed the xylem of the plant and can transmit the bacterium to an infected plant to a healthy plant. It is remarkable to mention that until this moment, the diseases that cause Xylella fastidiosa have no cure. So a collaboration between research groups is necessary in order to fight against these pathogenic bacteria. And that is the principal motive why the Versified network was created. The Ibero-American Network for the Surveillance of Xylella fastidiosa is coordinated by the Dr. Julie Peretó Mangraner. He is professor and research of the University of Valencia and Institute for Integrative System Biology at the CSIC, and he is responsible for the coordination of the network. Initially, he had to give this talk, but in the end, he has not been able to do it. So here I am. I'm going to try to do the best that I can. So let's go to start. This network is composed of more than 30 groups belonging to public and private sectors, including research centers, universities, and companies belonging to the agricultural sector. All these groups are from 10 countries, from Costa Rica, from Chile, Argentina, Spain, Portugal, and Italy. And the main objective of the Versified network is exchange information and knowledge, of course, between countries affected by this bacterium, by Xylella fastidiosa, in order, well, to generate new knowledge that can contribute to developing the surveillance system, which allow adopting the necessary measure to monitor, contain, and in a final state, try to eradicate the bacterium. Regarding the specific objectives that have this network, these are organized in different working packages. One working package is regarding the try to get information about the bacterium, paying special attention to the bacterial diversity, and try to create bacterial collection of isolate from the different countries affected by this bacterium. The second working package is try to get information about the vectors, trying to establish a catalog of the main vector insects from the different countries where the bacterium has been detected. Another working package is regarding to the plant interaction bacterial plant, focusing the study of the survival factor, multiply factors, and growing factor that have the bacteria when growing in plants. Another working package is try to get information useful for therapy try to control the bacteria, focus on the effective fight procedures against infection beyond to the plant eradication. And another two last working package regarding to the information about the teledetection method of this bacterium, and try to get information about the environmental, social, and economic impact of the disease caused by Xylella fastidiosa. Okay. And what is the methodology and working plan that we have in this network? Well, for this, we have different strategy and are carried out that along the exchange of information and knowledge between countries. One of them is the mobility program. This program stimulates scientific and technologies exchange between countries, of course, and also stimulate the development of predoctoral theses from different members of the networks or participants in the network. The network subsides two different mobility programs, one of long duration, up to 1 month, called training stage, and another of short duration, up to 1 week, called cooperation stage, okay? Well, I have to mention that as a result of the global pandemic caused by the SARS-CoV-2 virus, this mobility program did not begin until the last years. In the last year, in 2021, for example, 5 training stays and 3 cooperation stays were carried out, participating 8 different countries from Spain, Argentina, Mexico, US, Chile, Brazil, Costa Rica. And in this year, in 2022, so far they have 3 training stays and cooperation stays. I take this opportunity to tell the participant of the IberCFAS network to be encouraged to participate in this interesting mobility program. And another strategy which allows the exchange of information and knowledge between countries is the training program. This program consists in one way in preparation of dissemination of material addressed to the different sectors, private and public, of course, affected by the disease caused by Xylella fastidiosa, and developing press release, elaboration of extension article, organizing meeting and conference available in public platform like YouTube, for example. I think that this workshop is transmitting now in YouTube too. And another ways, organizing technical orientation, seminars, course, or workshop like this workshop, and forums. All of these forums addressed to the public administration and, of course, to the farmers affected by this disease. And final, another strategy which allows the exchange of information between countries is the program, the Extension and Dissemination Program. This program is addressed to all the social actors, of course, interested in the network, and it consists of the elaboration of monographies on a specific topic of the network, and in the organization of international conference and active discussion forum through the network internal web platform in theory, or using platform like YouTube or another similar platform. And I think that that's all. Thank you for your attention and enjoy the workshop. Thank you, Yao.
Thank you, Felix Moran, for the very important explanations about the activities of CITED and this important read of IberCFAS.
Thank you. You're welcome.
Very good. You can stay with us, and I will now ask I will introduce Professor Dirceu de Matos Jr., who will speak on behalf of Centro de Citricultura Sylvio Moreira. Welcome,
hello,
Dirceu.
Professor. Hello, Professor Lopes. Thank you for the opportunity to be here. It's a great pleasure to welcome all the participants and speakers to the Xylella Fastidiosa workshop. Wish to thank all the organizers for the efforts put on this event. Wish also to say that you will have the opportunity to meet outstanding people, as well find high quality information on this event about such important pathogen and all the diseases associated to that. For instance, in Brazil, by the end of the years of 1980s, the CVC, or the citrus variegated chlorosis, name given to the disease caused by this bacteria, was detected. And very soon, we accounted for serious problems in our citrus industry. we reached about 45% of the whole number of trees in our citrus industry infected by the bacteria and suffering with the disease. Estimates accounted for more than $140 million on losses, economical losses caused by the disease, mostly associated to the adverse impact that the disease caused on fruit quality and tree longevity in the field. With that challenge, research was conducted mostly on plant pathogen vector interactions. A new nursery production system was created. Now it's a reference for the world citrus industry, and today is producing about 15 million trees, nursery trees, healthy nursery trees per year. Also, with the event of the Xylella in our citrus industry, network for research and genome sequencing of the bacteria was established, and we came with the first pathogen in the world with the complete genome sequenced. Nowadays, Xylella fastidiosa diagnosis is mandatory in our citrus industry for nursery production. And if you recall the number that I gave you, that at that time 45% of the trees were infected, today we find less than 0.5% of infected trees. Then we have a great success on managing this disease in Brazil. So far, new insights on Xylella fastidiosa have been also found, what have supported new strategies to control other bacterial diseases. The knowledge accumulated with genome sequencing has supported this kind of strategy. Our research team has developed expertise interacting with people worldwide. Then hope you will find at the Citrus Center opportunities to visit and interact, especially with our fellow researchers, Sylvio and Alessandra Souza, and interact also with our staff in our laboratories and in our field. With that, I look forward to see you around, and thank you so much for the opportunity. Have a great time abroad or here in Brazil. João, thank you again so much.
Thank you, Dirceu. Thank you, Felix, representing the two main organizing and supporting institutions in this workshop. We cannot leave this opening session without acknowledging these organizing and supporting institutions, and also some members of the organizing committee, Alberto Ferreres, Alessandra de Souza, Carlos Chacon Dias, Francisco Beitia, Elvésio Coleta Filho, Joyce Froza, Juan Pedro Bouvet, Mariana Boss Esteves, Simone Prado, who helped us to build all the scientific program, and also technical support personnel who are helping us in this transmission, Fabiano Pereira, Regina Moraes, Euclides Villanova. We had a problem broadcasting the initial part of this opening session, but we will make available the whole opening session for you later. In order to avoid the delays, I will start the preparation for the virtual session 1. Speaking a little bit about how it's going to work. So we will have in this first virtual session 3 speakers with 30 minutes talks for each one. We are not going to open for questions during the talks. Instead, we are going to have the 3 talks, and then we will open for a debate where the questions will be brought to the speakers, and they can answer questions and they can debate about the topics they are going to speak. But the audience can put questions in the chat, in the YouTube chat, while the talks are being ministered, and we will take notes, and then afterwards I, as a moderator, will forward the questions to the speakers. There is a list of attendance. Some people are just watching, others are registered and want to receive certificates for that. So those that are registered and want to receive certificates, please fill in a Google form that will be presented in the chat about halfway in the virtual session, about 1 hour after, and you will receive the certificate if you attend at least 3 of the virtual sessions. Remember that we will have 5 sessions. This is the first one. So nearly every Thursday from 11 a.m. to 1 p.m. São Paulo time in Brazil. So let's move now to the first session. Today we'll have some good introductory talks about Xylella fastidiosa, its importance, about the classification, and about the mechanisms of interactions with plants. So the first speaker of the day will be Rodrigo Almeida, Professor Rodrigo Almeida. Rodrigo Almeida has a bachelor degree in agronomy by Universidade de São Paulo, a master's science degree in entomology, also by University of São Paulo, Brazil, and a PhD by University of California, Berkeley. Currently, he is a professor of emerging infectious disease ecology in the Division of Organisms and Environment at the Department of the Environmental Science, Policy and Management at University of California, Berkeley. Rodrigo is one of the major experts in biology, ecology, evolution, and management of insect-borne plant pathogens, especially on Xylella fastidiosa, where he has dedicated most of his career and has produced a large number of papers and articles and is recognized by his important contributions in this field. So, Rodrigo, it's already there. Hello, good morning.
Good morning. Can you hear me?
Yes, very good. So, Rodrigo, you are open to speak, and welcome to the workshop.
Okay, great. Thank you. Thank you, João, and for the organizers for the invitation. I do apologize, but it's a little bit early here, so my brain is not working as well. So what I'm going to try to do for this initial talk is introduce Xylella biology and a few topics that I think are relevant. And while I am doing that, I will try to also mention why certain aspects of the biology are of importance when it comes to disease emergence and quarantine and other issues, and hopefully finish on time and we can have questions later. So first, just want to acknowledge that we have received funding over the years from large numbers of institutions, and we have also a lot of very talented young scientists in the lab. This is a picture last week. Over half of these people are undergraduate students who really help do a lot of the work in our group at Berkeley. So we have heard and we know of many important plant diseases that have had large consequences to society or to the landscape. In the United States, here at the bottom, we have sudden oak death, which is a devastating oak tree disease. Of course, the Irish potato famine is something that led to devastation, a lot of death, as well as migration. And you have an example in France, for example. Here's a statue in Montpellier that highlights the challenges of phylloxera, which almost eliminated grapevines from France. and in some ways was solved by the introduction of American rootstocks. But we also have these sort of devastating diseases today. You have those that are historically important, and then you have some that are of significant importance today. So ash dieback in Britain is one introduction that has been very problematic, and the costs are estimated to be very high. So in this particular example here, they've estimated that 15 billion pounds as the cost of this introduction to the UK. So you do have plant diseases that are very significant in terms of impact to society. They are also very significant in terms of their costs. So let's talk about Xylella. So first, Xylella was first reported, not as a Xylella, but a disease caused by Xylella was first reported in Southern California as the California vine disease. And the person who wrote the first sort of scientific report on the topic was this guy over here, Newton Pierce. And he wrote a very interesting report, which he reads more like a book in some ways, where he described Pierce's disease, what we know today as Pierce's disease. He did actually a really good job identifying symptoms and other sort of patterns in terms of in the landscape and so on. But at the time he thought it was a virus, and which is something that remained true until the 70s when Xylella was shown to be a bacterium. So Xylella has been causing disease in California for over 100 years. And down here, there's a number that's more recent about estimates of costs to California that's suggesting that's $100 million per year. And I would argue that this is the cost of Xylella when we don't have a lot of it causing disease in grapevines. So 100 million in California per year is at, let's call it the low level amount of Pierce's disease in grapevines. At the high level, it would be substantially more than this. So here's a problem that has been going on for many, many years, and still causes substantial amount of damage, even when it causes very little disease. So Xylella is known to cause disease in several different types of plants. The host range today, estimated by the European Food Safety Authority, is about 650 plant species in many, many plant families. But historically, there's some crops that are well studied and known to be very susceptible to Xylella. So you have grapevines in the United States, mostly, but not only California, where it causes Pierce's disease. Historically, you have alfalfa dwarf, which is less of an issue now. Almond leaf scorch, which is always present in California, but at very low amounts, and now it's an emerging problem in Spain, for example. In the southeast of the US, funny peach disease, plum leaf scald is a problem in Brazil, for example. CVC, of course, in Brazil. And then you have lots of trees and ornamentals that are much less studied in the United States and in the Americas in particular. So we know that Xylella causes a lot of damage to trees on the east coast of the United States, but we know very little about it. And then you have ornamentals like oleander leaf scorch, which was a serious problem in Southern California, where it was essentially eliminated from much of the landscape. And then you have very large list of emerging diseases, and mostly coming out of Europe, olive leaf scorch being the main one. And but this list is expected to be growing, and it's certainly a subset of the plants and diseases that Xylella is actually causing. So while we're talking about the types of diseases Xylella causes, I want to emphasize that we know about Xylella diseases in agricultural crop systems. We know very little to almost nothing about Xylella in natural environments, where it certainly causes disease and affects the landscape, and also more urban, suburban, and rural environments where it's not connected to crops. So we know Xylella can infect all these different types of plants. There are reports of it, but we don't know exactly how many plants or what plants are infected and where, and we also don't know about the impacts. So this is a big gap about the importance of Xylella and the fact that we just don't know very much about it outside of crops, agricultural crops of economic importance. The other thing I want to bring up is that Xylella research has been driven by epidemics. So you can look at these different graphs here, but essentially what it shows, the bottom, they all show the same thing. This is publications per year with Xylella on the title or Xylella paper. And you can see here until 2000, when we had a large epidemic in California, the number of publications was relatively low per year. And then it went up here in the United States, mostly, but not only, research in California. And then this is also after when the genome was sequenced in Brazil, and research also went up. As you can see, there was no research from Europe. So Italy and Spain here, you don't really have any research going on until Xylella shows up in Italy in 2013, and that has gone up. And if you repeat this exercise today, you will see that Europeans are publishing at much, much higher rates today than Americans and Brazilians together, for example. So it's too bad it's driven by epidemics, because when the epidemic started here, there was no capacity in Europe. No one really worked with Xylella in Europe. So of course, there's a lag period until you actually get to know the system. And the same thing is really true at this point here in California and in Brazil. There were very few people that were actually working with the pathogen. So this is something to consider, because building capacity is important. So the biology of Xylella can be summarized in this slide, which is based on the olive disease in Italy. I'll talk about vectors in a minute, but you have insect vectors that are out in the landscape, and they will be infected with Xylella and feed on a plant and transmit Xylella to that plant. Xylella is xylem limited, and it will colonize the xylem vessels and migrate from one vessel to another through the pit membrane. Xylella infection leads to plant responses as well, and together between the infection and colonization and the plant responses, you end up with obstruction of the xylem vessels and leaf scorch or drought sort of stresses for most plants. I think Alessandra may talk about some of this later, so I'm not going to the details here. Once the plant is infected, you can have an insect vector that will come and identify xylem vessels and acquire the pathogen and transmit it to a new plant. And so this is an example of Xylella colonization of grapevines. So here's a little bit of symptom development in leaves. The interesting thing about Xylella is that you can have xylem vessels that are completely occluded, and right next to it, you can have xylem vessels that have no Xylella or very little Xylella. This is an example of what that looks like. Here's GFP labeled Xylella colonizing a vessel. The vessel is in red, the cells are in green. You can see that this vessel is completely clogged, colonized, and other vessels are not, right next to it. And what's really nice from this set of images here is you can also see cells crossing the pit membrane, and you can also see cells coming out at the other end. So the colonization sometimes can be a little bit patchy within the plant. Distribution may not be uniform, and that can also lead to challenges when it comes to detection of the pathogen. So one particular trait of Xylella is that it's insect-borne. It's only transmitted by insect vectors, and there are two groups of insect vectors, or two main groups of insect vectors. One are the sharpshooter leafhoppers, which are xylem sap feeders in the Cicadelidae subfamily Cicadellinae. Here are two examples from the United States. Graphocephala ultrapunctata here, which is native to the West Coast, native to California, and then Homolodisca vitripennis, glasswing sharpshooter, which is native to the Southeast, and now an invasive species in California. So these are they belong to two different tribes of sharpshooters. However, all sharpshooters are vectors of all Xylella until proven otherwise. Another group of insects are Spittlebugs. Here's Phylogeny primarius, the meadow spittlebug, which is the most important one in southern Italy as a vector today, and other parts of Europe. You have this insect as well as other spittlebugs that are vectors. There are different families of spittlebugs that transmit Xylella, and similarly to the sharpshooters, they are all vectors of all genotypes as well. So here's one particular trait of these insects is that they feed primarily on the mouth part, on xylem sap, or only on xylem sap. And xylem sap is under a lot of tension, so they have these huge heads with large muscles that are all dedicated for sucking sap. So here's what this musculature looks like. It's a little bit fast, but I hope you can see it. But all of this here up front in the head are muscles that, all of that are used to suck sap from the plant. So it's a pretty amazing musculature in the mouth parts here, these muscles over here, to suck sap, essentially suck 1,000 times the body weight of these insects at speeds that go through the food canal of over 1 meter per second. So we're doing some work now, which is using different technology. What you can see in blue here is essentially water that would be inside the head of this insect. So we're looking inside the head of the insect. And in this canal, this long blue tube here is the food canal. Here's where Xylella lives in the head of the insect, this little area here. And but sap will go through here at 1 meter per second, which is really impressive. And also necessary if you're going to ingest 1,000 times your body weight per day. Okay, so these are really interesting insects. Many of them are polyphagous, but in many ways they're only important from an applied economic perspective because they're vectors of Xylella. So there are a few important traits about transmission. One is that there is no vector species pathogen strain specificity. But this is kind of an interesting observation. But all insects in these two groups of vectors transmit all pathogen strains that have been tested. There's expectation that any pathogen that's introduced where you have a vector, any genotype of Zella will be transmitted by that vector species. So it's always important to demonstrate that the species can transmit it, but the expectation is that it will be a vector, not that it will not. So essentially, you have vectors everywhere in the world. So if you don't have Xylella and you have an introduction of Xylella, you probably have insects that can support spread. The question is if they will actually do it from an ecological perspective. The other aspect that's important here is that Xylella is persistent. So once an adult insect has Xylella, it will keep it for life. So already starting to run out of time here, but looking at Xylella ecology, one aspect that I want to mention is that we tend to think of a disease triangle, or what's called a vector disease triangle here at the bottom. But Xylella diseases and other vector-borne diseases are much more complicated. You have to consider time, you have to consider surrounding vegetation, multitrophic interactions, you have to consider the biology and ecology of the vector in addition to the host pathogen interactions. So everything really matters, and this makes for diseases to be much more complex. And in the case, again, of Xylella, you have this interesting phenomenon where you can have spread within your agricultural system, but you can also have spread within adjacent landscapes or other landscapes. And you can have spread from landscapes to your crop and from your crop back to these adjacent landscapes. So you can have different scenarios depending on the pathogen, depending on the disease, which in many ways is highly dependent on the biology of the insect vector. So you can have scenarios that are only here, and scenarios that link both environments. So you have these layers of complexity that link that are important, including environment, temperature, for example, we know is a really important factor for Zalala. Then you have the ecology of the different organisms involved in the system, the outcome of these interactions, and of course, disease management is something that will also determine how much disease you're going to have over time and how well suppression approaches are working. And that's all just to say that management strategies for Zella diseases have to be disease specific. There is no one sort of approach that will fit for everything. The best example is Pierce's disease of grapevines in California, which is essentially one disease in the same state. And in Southern California, because you have one vector species, the management strategy is completely different than in Northern California, where you have a different vector species. So it's really important to remember that you can have the same disease, same name, even if it's in the same country or the same region, like in our case, and the management ultimately may be very different for different sort of factors. It can be the insect vector species, it can be the landscape, temperature, and other considerations. So I want to mention that Zalala also has a very dynamic genome. It has been shown to change in many different ways. This is something that will be talked a little bit later in focused on diversity. Zalala can acquire genes in different ways. It can exchange genes through, it can acquire genes through transformation, just acquisition of DNA from the environment. It can also have phage infecting it and leading to change. And it's also subject to conjugation. So the genome is fairly dynamic. It changes quite a bit. And particularly transformation and homologous recombination have some relevance and have to be considered when we're thinking about taxonomy and quarantine of the pathogen. I will skip this one here due to time. So the historical sort of phylogeography of Zalala is that it was a pathogen of the Americas. This is sort of what I've learned when I was in grad school. You have subspecies Palca in South America, Multiplex in North America, and Fastidiosa being originally from Central America. So these are historical ranges for these different groups of Zalala. Of course, taxonomy is something that it's arbitrary, but these are three groups are well supported, and these three groups do have sort of this geographic distribution in terms of the areas that have the most diversity. But now we know that Zalala is essentially everywhere. And these are some routes that of dispersal that we have identified based on genome sequencing and others as well, not only us. What's interesting here is that we still don't know a lot about Zalala for much of South America. We know it's in most places, but we don't know exactly where. And we don't know what the genotypes are. The same thing is true for Central America. The United States is the best studied region, except that it's mostly focused on crops, as I mentioned earlier. In the last 10 years, there's been a lot of interest in Europe, Southern Europe, where Zalala has been identified in multiple places. Recently has been reported from Israel, and then as well in Taiwan. What I want to bring up is the fact that you can see that there is no Zalala reported from Africa, no Zalala reported from Asia, except for Taiwan, and also Australia and New Zealand. And it's really hard to imagine that you don't have any Zalala anywhere here. Australia and New Zealand is a little bit different. They actually do a lot of surveillance, and these are isolated regions. But much of these over here, as well in Africa, it's likely to have Zalala. I wouldn't be surprised if there are reports emerging over time. An important thing is that while we know of all of this trade here that has led to spread of the pathogen, we also know that much of these emerging diseases and much of the dispersal of Zella happened in the last century, and we assume that most of it was because of the trade of contaminated plant material. So most of these epidemics in Europe, not most, all of them, as well as some other introductions like in Taiwan and Israel, for example, different methods have been used, and they all indicate that these are early, are young diseases. They are young introductions, and they all come back to the same period during the last 50 years or so. And the assumption is that these were all contaminated plant material introduction or based introductions that we're finding about them now only because we have started to survey. These are not pathogens that were introduced and plants that have been sick just during the last few years. They've been infecting in plants, particularly in Europe, for decades now. So because of concerns in Europe, we have, the community, have been thinking about issues of Zella host specificity. So many pathogens, you know what drives specificity. In the case of Zella, we do not. So my group, as well as other groups, have been looking at the fact, what drives host specificity in Zella. So that's, trying to find out if one particular genotype causes disease in one host plant and not in another. So we've looked at trying to look at patterns of co-speciation, for example, which would help us understand a little bit about the evolution of the pathogen and the host plant, and we see no patterns. And long story short, we've been trying to understand a little bit better this issue of host specificity, and it's still very complex, and we're making headway, but we still don't really understand it very well. I want to bring up that this is important. It's an important topic because in Europe, in particular, guidelines to manage the disease, to manage the pathogen, particularly in terms of eradication and quarantine efforts, at least were until recently, I think they currently still are, based on the subspecies level of the pathogen. The problem with that is that subspecies level is really not helpful in terms of inferring the host range of a particular pathogen genotype. So eradication efforts are going on that consider all the potential hosts of Zella, but we know that there is a lot of errors that are built into this sort of subspecies level definition of host range. And understanding the host specificity better would allow regulators to better define which plants are potential hosts of a pathogen in a particular location and be more specific when it comes to eradication efforts and trade as well. So I want to very briefly just mention one particular new disease, not so new anymore, but it has been particularly devastating, and that's the problem of Xylella in olive trees in Italy. The disease was first reported in 2013. It has been all over the news. It is the problem that has triggered the formation of this particular group here that's leading this workshop and has triggered all the research in Europe that we see today. So it's a problem and it has been on the news. It's a particularly devastating disease. You can see here a photo of an orchard with some symptoms in 2013. Just 3 years later, the plants are essentially dead. The disease has been moving up north. It has moved farther north now. And there is really no solution in sight other than folks are trying to plant what looks to be tolerant plant material here. And hopefully that will be a short and long-term solution for the local olive industry. The point I want to make is that knowledge has been accumulating the last few years. There's a lot of funding, there's a lot of research. So people are understanding better the ecology of the vector and how that leads to spread. There's a lot of modeling of different types to look at the spread and risk factors. There's a lot of work on the pathogen itself as well, looking at genomics, but also the biology of the pathogen, host range, plant resistance. And this has been really great because it has really helped understand what's going on. I also want to bring up that in Europe, you have meetings about Zalala essentially all the time, and research capacity is being built quickly. The point I want to make about the research capacity being built quickly, again, is that 10 years ago, literally 10 years ago, there was essentially no lab in Europe that was working with Zalala other than being able to do some detection. So everything in Europe is less than 10 years old, it's 9 years old today. And but it's, you know, this is changing very quickly. But in other regions where you still don't have Zalala, it would be really important. Or if you don't have capacity, it would be important to build this capacity as soon as possible before you have a serious epidemic. So the point I'm making here is that building academic research takes time, and if you don't have research capacity in place, you have to develop that research capacity. And in many cases, especially in Europe, it's important to remember that the technology-based solutions may not be sufficient by themselves. So I have a quote here from a farmer that I want to read that highlights some of the challenges on the ground. So this farmer is in the region like this, where you have a lot of dead trees. By a lot, I mean millions. And here's what he had to say. I had to sell the olive mill to let it become wreckage, okay? And I already had to sell it for nothing. I didn't earn anything last year. I won't earn anything this year. So this person has really lost money and is not sure what to do. And then he asks, how can I do the replanting myself with what income? Then once I have replanted, how do I water it? I have to connect the water well. I have to pay for electricity. How do I live then? How do I go on? So even in this case, he's alluding to resistant plants. Even if you have resistant plants, it takes years for these plants to produce a crop. There is a huge cost in keeping those plants alive. And how is that individual going to do it? So the point, again, is that even if you come up with a solution, how do you actually implement it? It has to fit into a broader socio-economic context, and it's something that us as plant pathologists really do a poor job of understanding how to do that and how to implement these strategies in a situation like this. So let's just skip this, but there have been some drama in Europe. I want to point out that Zalala now has been found all over the Mediterranean basin, and many of these are independent, different introductions. So it has been introduced many times, just it was there and people had not found it yet. And much of this, there's a lot of evidence suggesting that is through imported plant material. So here's some kind of examples. The volume of plants, the number of plants traded or imported in Europe from plants that are in lists of hosts of Zalala in a period of 7 years was 158 million plants. And this is plants for planting. It does not include certain types of plants here. So there's a lot of trade, and if you're importing plant material from regions that have Zalala, there is an expectation that some of those plants will be positive. So I want to just finish by saying that Zalala is an agricultural threat. That's how we understand it. But diseases caused by this pathogen also impact the environment and society. And those are aspects of Zalala, its relevance and its risks and its importance that were really poorly understood. And that's something that should change, or I hope will change. But so far, we're really only focusing on one particular aspect, and that is really an issue if you think about it in the long term. So with that, I will stop sharing and thank you all and take questions when time comes. Thank you.
Thank you, Rodrigo, for the very nice talk. Very nice overview, comprehending several aspects related to Xylella fastidiosa. And now we are going to move, as I said before, we are going to leave the questions for the end. and we are going to move to the next speaker. Now we are going to call Alexandra Kahn. Alexandra is Bachelor in Environmental Studies by Oberlin College in Ohio. Currently, she is a PhD candidate in Environmental Science, Policy, and Management by the University of California, Berkeley. She works at Rodrigo's lab, and she has very good experience on genetic diversity and taxonomy of Xylella fastidiosa. So, Alexandra, good morning.
Good morning. Yeah, thank you so much for the invitation. It's really wonderful to be here.
You are welcome. Feel free to start your talk. Thank you.
Okay. All right. So today I've been asked to talk about genetic diversity and taxonomy of Xylella fastidiosa. So taxonomy is often perceived as a bit of a curmudgeon-y science with old museum records and boring papers and things like that. So I'm going to try to integrate a little bit of applied and functional work into the taxonomy so that it doesn't get too dull. But I want to make sure that we get situated in the genetic and taxonomic world of Zella. So to start out, I want to just give a brief introduction to phylogenetic trees to make sure that everyone is on the same page. I'm going to be showing quite a few phylogenetic trees throughout this talk, and it would be a shame if anyone doesn't really understand what's happening. So I just wanted to start with a good definition of phylogenetic tree that I like to think about and use whenever I can, which is that a phylogenetic tree is a visual hypothesis of common descent. So trees are a very natural way to visualize biological relationships. And that's because, to quote authors I like, Baum and Smith, the evolutionary history of species nearly always involves the branching of evolutionary lineages without subsequent fusion. Of course, as probably many of you know, Xylella fastidiosa is one of those cases where nearly every time, nearly always, biology is proven wrong, because we actually do see branching with subsequent fusion. But we'll talk about that more later, and just for now, assume branching. Okay, so I'm going to give a little bit of the anatomy of a phylogeny. So every time you look at a phylogenetic tree, you're going to go from narrow to broad. So as you go from that narrow to broad direction, you're going either through time, if it's a time-calibrated phylogeny, or if it's the case of a typical phylogeny, usually it's made with genetic material, with DNA, and you're using the axis is actually a substitution rate. So it's the accumulation of substitutions from mutations across the phylogeny. And then we have, in these branching points, they're called nodes. So these represent a hypothetical ancestor, not an actual data point. Whereas the tips here represent the data points that we have inputted into the study. And that could be a genus, species, or just some strain that some person found in their backyard. And then finally, we have a clade, which is an ancestor, so a node, and all of its descendants. To be a proper clade, it needs to include every descendant. It can't just pick and choose some of them. So when we have a clade like this, with these A, B, and C taxa, here we have A and B are more closely related to each other than either one is to C. So in a tree like this, it can actually be left to right or right to left, doesn't matter. But an important thing to remember is that only these horizontal distances matter. So these are the branch lengths that show how many mutations you've accumulated. But these vertical branches, they don't represent anything. They're just a way to spread out the data, which also means that any node can be flipped top to bottom. So on the left, we have ABC, and on the right, we have BAC, but these are the same trees. So this is the most common mistake that people make when looking at phylogenetic trees, and I want to really hammer it in clearly, that just because B and C are physically closer on this screen to each other does not mean they're closer related than A is to C, because you can swap this node, swap. Okay, so when we think about Xylella, we actually see a very similar 3-branch tree that's set up in a similar way. So we have subspecies Pauca, which could be our C, and then subspecies Multiplex and Fastidiosa. So Multiplex and Fastidiosa are more closely related to each other than either is to subspecies Pauca. However, just because they're laid out in this order does not mean that Fastidiosa is closer to Pauca than Multiplex is to Pauca. So we can switch it, and it's exactly the same tree. So make sure every time you look at a tree, you're tracing back the nodes and not just looking at what's next to each other in a list of names. Okay, so if you take away anything from this, hopefully it's that. All right, so in terms of the history of what we know about Xylella fastidiosa phylogenetics, as Rodrigo mentioned, in California, it was first reported in the 1880s, but it wasn't until 1987 that Wells and al. Named the genus and species of Xylella fastidiosa, because it took a very long time to even realize that it was a bacteria and not a viral disease. So in 1992, Chen et al. Show with RFLP 2 distinctive groups of Xylella fastidiosa. And then in the year 2000, we have the honor and privilege of Xylella fastidiosa being the first whole genome of a plant pathogen that was fully sequenced. So not until 2003 were the 3 subspecies of Xylella fastidiosa defined, Pauca, Multiplex, and Fastidiosa. And then in 2010, we created an MLST set for Xylella. So using 7 housekeeping genes, a type of barcoding system was implemented that is still sometimes used. And then in 2019, the first whole genome phylogeny was published, including 72 strains of all 3 subspecies. And since then, there's been a huge increase in sequencing effort, and we're up to over 400 or almost 500 whole genome sequences of Xylella fastidiosa strains that are accessible on NCBI. So let's get situated within the greater bacterial realm. This paper is focused on Xanthomonas. So Xanthomonas is a bacterial genus that's typically considered to be closely related to Xylella. So Xanthomonas, they're typically also plant pathogens, but they generally have an environmental phase and are able to enter plants without a vector, unlike Xylella. So they cause various diseases, including blights and bacterial spot diseases. And Xylella is typically described and distinguished compared to Xanthomonas as having a reduced genome, substantially reduced genome compared to Xanthomonas. Xylella also lacks a type 3 secretion system, which is characteristic of Xanthomonas. Xylella is insect associated, Xanthomonas is not, as well as Xylella being xylem limited, which only one of these various Xanthomonas species, Xanthomonas albalinians, is also xylem limited. However, when we look more closely at this tree, we can see this blue clade 2 is 3 strains of Xylella, including both Xylella taiwanensis and Xylella fastidiosa, which are the 2 species in the Xylella genus. But Xanthomonas is both up top and down below, which means that in all of the research that we've been doing and saying, oh, Xylella and Xanthomonas, they're neighbors, they're closely related, that's not really accurate. In fact, Xylella technically is a member of the Xanthomonas genus. Either that, or Xanthomonas is just like kind of a concatenation of multiple genera that needs to be split. So in terms of the Xylella research, frankly, I don't think it really impacts things too much because it's such a robust system and there's so much work within the species. But if I was working on the Xanthomonas diversity, I would be a lot more concerned. So like when you look here at these Xylella strains, they're more closely related to all of these strains in green up here, which include kind of pretty famous Xanthomonas like Xanthomonas oryzae, which infects rice, and Xanthomonas campestris, which causes blight and all sorts of cruciferous vegetables. So Xylella is more closely related to them than they are to Xanthomonas albulinians or Hyacinthi or other ones over here. Yeah, so Xylella is still unique in all of these ways, but it has to kind of function within the Xanthomonas genus. And throughout the rest of the talk, I'm gonna just completely ignore Xylella taiwanensis, just in case anyone is curious. It's the other species, but there's been a lot less focus on it. But it infects pear in Taiwan. Okay, so this is a tree that you saw in Rodrigo's talk as well, and I'm kind of going to use it as the backbone for this talk. So you're going to see this tree again and again. So this is a time-calibrated phylogeny. So I said that most phylogenies are going to be just based on substitutions, but this one, the authors calculated the typical Xylella mutation rate and were able to convert that substitution rate into estimated time. So you'll see that they're pretty broad time estimates, but they can give us a basic idea of how long ago these different strains, subspecies, and groupings of all sorts have split from each other. So you see that the estimation for the time of the overall split of these three subspecies is about 10,000 BC, so 12,000 years ago. So we don't have an estimate for what the native range would have been for that ancient ancestral grouping, but we have our three subspecies, Subspecies Pauca, Multiplex, and Fastidiosa up top that are have been diverging since then. And Subspecies Pauca is known to be native to South America. Most of the work has been done in Brazil. Subspecies Multiplex is native to North America, predominantly in the southeastern United States. And Subspecies Fastidiosa is native to Central America. And the vast majority of the strains from Central America have come from Costa Rica, where there's a very robust research infrastructure for Xylella. So I'm going to talk about one subspecies at a time, really. And I'm going to start with Pauca. So we can see that there's a big split right here, about 3,000 years ago. And so Pauca has at least two. This kind of one on its own is a little bit more ambiguous, but I'm going to focus on this more robust split. And you can see little pictures of maps next to them. So this split delineates the strains that are in both Costa Rica and Italy from the strains that are in Brazil. So when we think about the emergence of subspecies Pauca in Europe, as well as in Costa Rica, we think of it as, I mean, for me at least, I think of it first as, oh, it's subspecies Pauca is from South America. However, it's been over 3,000 years since the subspecies split. So the introduction into Italy is from a lineage that is long diverged from what we now see in South America. Okay, and then generally about subspecies multiplex, it's a quite young split compared to the others. And it generally is defined as having two groups, and one is kind of just an older group and one is a newer group. And then for subspecies Fastidiosa up in blue, we also have two main lineages, and I'll go into them more later. But one of them predominantly is still in Central America, and another one is split between Central America and then all of the outbreaks that we've seen in other places. And then in black here, we have a couple of other lines that have been kind of neglected, and they're considered subspecies Morris and Sandii, which are not as clearly defined or supported as the other three subspecies. So they're kind of like the potential subspecies that might be, whereas these three are really strongly reinforced as subspecies. Okay, and then, so Pauca, the most important hosts that we think about, I've kind of placed next to them so we can trace it throughout the rest of the talk. So for subspecies Pauca, the three most important hosts are typically citrus, olive, and coffee. Subspecies multiplex is one that if you're interested in forestry, you might have an interest in focusing on, because it infects many trees, including oak, elm, sycamore, hickory, and then agriculturally it infects lots of prunus like peach and plum, but also blueberry and other non-tree species. So Subspecies Fastidiosa, some of the main outbreaks that we've focused on have been coffee and grape. So every time you see one of these little logos, you can try to remember which subspecies we're talking about based on the host. All right, so subspecies pauca. So I've zoomed in on a more clearly defined split that I was talking about before between the strains that are located in Costa Rica and Italy versus the ones in Brazil. And we also, most trees that are made of subspecies pauca, you see that this Hib4 strain, which is from hibiscus, is often the outgroup. So, and it's the most different from all of these other Brazilian strains. So if anyone is ever interested in focusing on this one strain, I think you could find some interesting work. But there's a lot more diversity, a lot longer branch lengths between these strains in Brazil than there is in Costa Rica and Italy, which shows that these strains have been diversifying for a much longer time than these two recent outbreaks, even though this split might have happened thousands of years ago. So I guess that leaves a question of if there are other strains in this top clade that we're not aware of that might exist, that upon further sampling, potentially in Costa Rica or in that big vast majority of South America that Rodrigo mentioned had been kind of undersampled. I would not be surprised if we found other strains that fit in between these two clades. And this is another figure from Donegan et al. That shows some of the strains from Brazil. So this is work in progress. So it might change a little bit, but you can see that there's a delineation of clades where citrus and olive are the hosts. And then you can see coffee is kind of scattered throughout. But within Brazil, you have multiple hosts, but you do have genetic groupings that kind of delineate hosts. So as Rodrigo was saying as well, you can't necessarily base a host on a subspecies because there are much, much smaller groupings that are going to delineate host. However, just because you've isolated a plant from a host doesn't mean that it can infect other plants. However, and this is one of the few systems in which there have been robust cross-inoculation studies showing that citrus and coffee don't actually co-infect. Okay, so now talking about Subspecies Fastidiosa. So this is a tree from Castillo et al. 2019, where you can see the split of Subspecies Fastidiosa. So looking back from the root where it connects to multiplex and the other subspecies, you can see this first split, and then in the bottom clade, all of the strains are in Costa Rica. So this is the native range where the most diversity is found. And then in the upper clade, you have 3 strains from Costa Rica, and then you have strains that have been introduced everywhere else. So you have Texas, California, Spain, Florida, Taiwan, and I think Israel has been added to this more recently. So all of the global outbreaks stem from this one clade that has a deep split from the other clade. So California Pierce's disease is one of the most intensively studied systems. And because of that, there's been a huge amount of sequencing. So in terms of a subspecies, we have these big triangles, just to go back for a second. And those can represent hundreds of individual strains that have been sequenced. And it might seem like overkill to do that, because why wouldn't we sample all across the world and try to get more hosts and a greater understanding of diversity? But there's actually a lot to be gained from focusing on one narrower area. So just across California, so from Temecula to Sonoma, this is just kind of the southern 2/3 of California. You can see that just since the introduction in the 1880s, there's been a lot of population structuring based on geography. So you can see that these different regions have isolated from each other and diversified. And that's reflected in things like adaptation to the environment. So because those strains were able to be compared to each other, and because there were so many, there's a lot more computational power. And for this particular study, it allowed the authors to make correlations between environmental variables and different single nucleotide variants. So in this slide here, you see 6 different single nucleotide polymorphisms that are each associated with a different bioclimatic variable. So some of them are based on precipitation, some are based on temperature. I think it splits like cold temperature versus hot temperature, and also based on altitude. But if it weren't for the high density of sequences in one region of one subspecies, we wouldn't be able to make these connections. And in recent studies that are not yet published, it seems like temperature range, even within California, does have an effect on whether a strain can survive the winter in a northern location versus a southern location. Okay, so now let's talk a little bit about Subspecies Multiplex. Native to the southeastern United States, one of the biggest historical focuses of Subspecies Multiplex has been phony peach disease, which I just wanted to say because it's my favorite Xylella disease name. Shouldn't really pick favorites, but calling something phony feels it's just great. Not that I'm advocating for the disease. I'm definitely on the plants team, but okay. So in the previous subspecies, we talked a little bit about how trees could help us understand environmental adaptation. And in Subspecies Multiplex, authors here took a different route. So they were interested in trying to understand the history of introductions of Subspecies Multiplex into Europe as well as into other regions. So because they have a robust phylogeny, you can see high bootstrap support at the branches, at the nodes. They were able to do an ancestral state reconstruction to understand the history of the geographic origin of each node. So the ancestral range of this subspecies is the southeastern United States. So you can see at different nodes, there's predicted a high likelihood of this node being present in the southeastern United States as the origin. So the question really is, do we have one introduction into Europe and then a diversification within Europe? Or did we have multiple introductions into Europe at different points from different parts of the tree? And so with this tree here, you can see from this node originated in the southeastern United States. And from there, it goes to Texas, to Brazil, to Georgia, to California, and to Tuscany in Italy. And then you have another node up here that's also in the southeastern United States. And from this node, we have strains that go into Spain, California, Georgia, France, and Mallorca, just different part of Spain. So you can see that based on this phylogenetic grouping, which, just looking at the trees is not necessarily fascinating, but once you see how much power you can have in terms of understanding the adaptive potential and also the geographic history, it can be pretty useful. Okay, so I want to talk a little bit about our two outliers, so to speak, Morris and Sandii. Morris typically infects mulberry, while Sandii often is known to infect oleander, but there have been some hints about potentially also infecting coffee. So in these 4 trees to our left, they're all whole genome, based on a whole genome sequencing of about 350 Xylella fastidiosa genomes, but they were made using different parts of that alignment. So the top right here, we have our core genome tree. And for this tree, you can see our subspecies Pauca, subspecies Multiplex, and then Morris and Sandii here. Those are MUL-MD, are members of Morris, and N1 and R8 are members of Sandii. So you can see that these two are kind of the outgroups to Subspecies Fastidiosa. And this is the same topology as what we saw in the Van Hove tree that we've used throughout the talk. It kind of fits our expectations and it makes sense. However, we also have a tree here where recombinant regions are removed from that alignment. Zella has a lot of recombination, given as it's naturally competent. And when we remove the recombinant regions from the alignment, the locations of these two ambiguous subspecies move. So Morris is now part of Subspecies Festidiosa. It's within it. And then if we change it again and use the MLST genes, so those 7 housekeeping genes, we have another swap. So it switches completely on its end. So now we have Morris is the outgroup and Sandii is within Subspecies Fastidiosa. And finally, there's a pangenome tree, which actually looks a lot like the core genome tree. This is just based on presence and absence of genes. However, Pauca gets all split up. So really what we're seeing here is both that Morris and Sandii are kind of mobile, depending on recombination, and also that our MLST alignments don't look the same as our core genome alignment. So they don't recreate the same topology. And so you can see here, this is a recombination plot, and the stark blue regions are Morris and Sandii, and they're just rife with recombination. I'm not sure about the percentage, but I think maybe half of the genome of each of these subspecies is due to recombination with Subspecies Multiplex and Subspecies Festidiosa. And this is not unique to these subspecies. Zella in general has very large swaths of recombination in all of the subspecies, but Morris and Sandii are especially prominent. Okay, so aside from the MLST issue that I presented before, which is that the topology doesn't match the topology of our whole genome data, it was showed in Landa et al. 2019. I'll just read this quote. While MLST is particularly powerful for the taxonomy of recombinogenic taxa, it is also possible that strains belonging to the same ST may not be phylogenetically related or share similar traits, such as host plant range. And this was showed in this paper because there was a sequence type that was being focused on, and it was actually 2 lineages in 2 places, but they had the same sequence type. So it's phylogenetically, it's a reason not to focus on sequence types, but they also aren't really definitive of or descriptive of a species. And so this is, Rodrigo showed this image as well, but this is an ancestral state reconstruction of host history based on the whole genome of 350 Zella fastidiosa strains. So you can see our 3 subspecies. Subspecies Pauca. Just to get oriented, here's our Brazil clade, and here's our olive Italy and Costa Rica clade. And then we have Subspecies Multiplex over here. And then up top, really the whole half of the top is Subspecies Fastidiosa. So what this tree shows is the color of each node is mapped as the most likely ancestral host. So for Subspecies Pauca, at that big split that was several thousand years ago, there's not like a hypothesized host, but you can see that there's a lot of switching back and forth between coffee and citrus within Brazil, and olive as well. However, for Subspecies Multiplex, you can see at a lot of the ancestral nodes that blueberry and also the genus Prunus, so Vaccinium is the genus for blueberry, are very prevalent hosts at ancestral nodes. So these splits that were several hundred years ago. So the splits that we have for multiplex are much more contemporary than that for Pauca. And that enables us to see that some of the likely hosts in the last several hundred years have been these 2 genera, Prunus and Vaccinium. Which makes a lot of sense given that those are both native plants, or those genera have many native species within the southeastern United States. And then for Subspecies Fastidiosa, if you focus on this node here, this is the node that's ancestral to the whole subspecies. Here we have Costa Rican strains, and here we have California and other outbreaks. But this node here, that's the ancestral node to the outbreak of Pierce's disease in California from Central America, which was hypothesized to have happened a couple 150-ish years ago. And it shows here that the most likely host of that introduction event would have been coffee. which has been hypothesized in other areas too. So using this phylogeny, we're able to make predictions about the history, and hopefully that can help us make decisions about how to move forward in the future, what hosts might be particularly susceptible, what hosts might be more likely to spread the pathogen based on trade. So just very briefly, just to reiterate, these are very agriculturally focused strains, but Xylella has been evolving for thousands of years in natural systems. So if we really want to understand the taxonomy and diversity better, we need to go sample in more of these native systems. All right, so thank you so much to my lab. Thank you for having me today.
Okay, thank you very much, Alexandra, for this very nice talk. It's amazing how diverse is Xylella fastidiosa. Within a single species, we have such a variety of genotypes, and so it is very important to have this information in order to understand all the biological and ecological aspects. So thank you very much for your talk. Let's wait for the questions, and now I will introduce the next speaker. Thank you. We are going to move to the next speaker, which is Alessandra de Souza. Alessandra is a biologist, graduated from Catholic University in Pernambuco, Brazil, has a master in microbiology by the University of São Paulo, a PhD in genetics and molecular biology by the State University of Campinas, Unicamp, did a sabbatical at University of California, Berkeley in 2009. She is a researcher, scientific researcher at biotechnology in the Citrus Center, Sylvio Moreira, and she's an expert in the fields of genetics and molecular biology, with an emphasis on phytopathogenic microorganisms and their interaction with the host plant. So, Alessandra, welcome to the
Hi,
workshop.
John. Good afternoon.
Our audience, yes.
Okay, thank you very much. Thank you very much for the invitation.
Start your...
Okay, I think that I'm going to open my presentation. Let me see here. Can you see, Simone? João, it's open, my presentation. I can't see you.
Alessandra, it was perfect.
Okay, I couldn't hear you. Sorry. That's why I finished. Yeah. So I'm going to start again. Please let me know if can you see me, please.
Yeah.
It's okay?
Cool. Yeah, no, it's perfect. Thank you.
Okay, thank you very much. And I'm sorry for this delay. And so firstly, I would like to thank again for the organizer for the invitation to talk about Xylella fastidiosa, mechanisms of pathogenicity and host interaction. So as Rodrigo Almeida already mentioned, Xylella fastidiosa is a multi-host pathogen because it causes disease in many important crops such as citrus, olive, plum, almond, grape, and many others. So during my presentation here, I'm going to focus on the citrus-Xylella interaction, but also I'm going to point out some example of conserved mechanisms that occur between Xylella citrus, but also with grape and olive. Okay, regarding these three crops, it's very common. I mean, you can see in many papers that when the symptoms occur on the leaves, in that case, is Pierce disease on grape, olive quick decline syndrome on olive, and citrus variegated chlorosis or CVC on citrus, is possible to observe a huge colonization of Xylella fastidiosa in the xylem vessels. And this colonization is systemically, I mean, the Xylella is able to move through the xylem vessels and then systemically colonize the host. And this colonization, this ability to colonization and survive in the xylem host is considered the main mechanism of pathogenicity. So, just to emphasize this, the capacity of the bacteria to move and colonize the xylem vessels is considered the main mechanism of pathogenicity. But how it occurs? It occurs in two different phases. So Zylella fastidiosa, the lifestyle of Zylella fastidiosa is closely related with the ability of the bacteria to grow on the biofilm formation that is considered the sessile phase. I mean, the bacteria is not move when the bacteria is in the biofilm. And the other one is the planktonic phase, or the step or the phase that the bacteria can move and detach from the biofilm and move through the xylem. Both phases are very important to the lifestyle of Xylella because this one is important to the bacteria can survive to a stress condition. And the other one is important to the bacteria move and find another environment to grow. So that's because the importance of these two phases, there are many works, many manuscripts already available that shows that genes involved in this phase or genes involved in this phase, when it is mutated, the pathogenesis of the bacteria is affected. So one gene or other one, if it's mutated, directly affect the capacity of the bacteria to cause disease. All right, but what regulated the genes? How the bacteria can know if the bacteria needs to be in biofilm or move? It is regulated by choron sensing. So choron sensing is a mechanism of cell-cell communication that in high cell density, the molecules of choron sensing, that in the case of Xylella, the more known molecules associated with the choron sense is DSF, diffusible signal factor, that is synthesized by RPFF enzyme that is encoded by one gene with the same name, RPFF. And when the bacteria is in high cell density, these molecules is recognized, is sensed by Zylella fastidiosa, and positively regulated genes involved with the biofilm. So in high cell density, high DSF, no movement, sessile phase, biofilm formation. And in low cell density, I mean, in the DSF, negatively regulated genes associated with the movement. It means that the SF positively regulated genes involved with biofilm in high cell density, but negatively regulated genes associated with the movement. And why it is important? Because it's the regulation between one gene and the other that the bacteria use to colonize the xylem vessel. And this high colonization is associated with the mechanism of pathogenicity that we can observe these symptoms of the disease on the plant. And to prove that, the team of Dr. Steve Lindahl from UC Berkeley, using grape, in that case is the grape model, they use a mutant, a bacteria, Xylella fastidiosa mutant to RPFF gene. And this mutant completely change the regulation of the genes associated with the biofilm and movement. And they observed the opposite. Without DSF, the bacteria move more faster in the xylem vessel, and consequently, it was observed more symptoms. So they raised the question if could plant expressing RPFF affect bacterial signaling? The idea was if they use one gene from Xylella and transform plants, could this plant be more resistant to Xylella because we will confuse the bacteria and then the bacteria will not express gene with movement but express more biofilm and then will not move? So the team from Dr. Lindahl showed that in one paper in 2014, where they used one gene from Xylella fastidiosa, Subspecies Festidiosa, that causes Pierce disease, transform grape, and DSF produced by transgenic plant confused the bacterial signaling, and consequently was more biofilm, the bacteria did not move very well, and so occurred the reduction in severity of Pierce disease. And so, in collaboration with Dr. Steve Lindahl, we started to work on citrus to see if this approach used for grape could be also used for citrus, if this mechanism could be conserved between both hosts in relation to Xylella fastidiosa pathogenicity. So, we use the gene from, but in that case, from Xylella fastidiosa that causes CVC disease, not grape disease, because it is Xylella fastidiosa subspecies pauca. And then we use these genes, we transform tobacco as a model plant and also citrus. We always use the tobacco to analyze the function of one gene or tobacco or another model plant like Arabidopsis to first verify if the hypothesis is applied to model plants. And then we transform citrus because the citrus take a long time. It's very hard and difficult to get transformed plants. And so after we have to propagate it, and then we have at least 3 years to have plants already done to be challenging. That's why we always use tobacco as a model plant. And so we got good results with tobacco, and then we move it for citrus, we transform citrus, and we verify similar results observed with grape, because the Xylella was not able to colonize efficiently citrus, the transgenic citrus plants, compared with wild type. If you observe it here, the Xylella movement, the Xylella was not able to move efficiently in transgenic plants compared with the wild type. And so when we we analyze the gene expression of Xylella that grew on transgenic plants, it related to Xylella that grew on wild type, we verify that the bacteria expressed that grew on transgenic plants significantly expressed more genes associated with biofilm formation and repressed genes associated with movement. And consequently, without colonize or move efficiently, we observed less symptoms. In that case, the transgenic plants, Hamley varieties, and the other varieties of citrus, sweet orange, pineapple varieties, we can observe much less symptoms of the transgenic plants compared to the wild type. Here is the score just to compare. In transgenic plants, the most symptoms were only the score 2, between 2 or 3, and this or this, while in the wild type we got score between 4 and 5. In this picture, you can see a better representation of the symptoms we observed. In the wild type, we observe a much more severity of the symptoms when we compare with RPFF transgenic plants. The same one to other citrus, the sweet orange varieties, much more severity of the wild type when we compare with the transgenic plants. This paper was published in 2017, but if you want to see the details of this study, I invite you to read this paper. But what I'd like to point out here is this conserved mechanism, because Xylella fastidiosa pauca and Xylella fastidiosa fastidiosa that cause Pierce disease, both use this mechanism to colonize the host, and this colonization is directly involved with the symptoms. So if we can decrease this colonization, use methodology or strategies like we use it here to impair this colonization, you can decrease the symptoms of the disease. So nowadays, these plants are evaluated on field condition, because besides resistance to disease, we need also to observe the agronomic traits to see if in the future, not now, but in the future, this plant could be used for citrus production. But now it's only research study. And so these plants are on field condition. We observe the similar weight of the wild type and transgenic plants, the same for photosynthesis. And then we also developed an antibody against RPFF, the protein, And we localized this protein in the transgenic plants. And so we observed that RPFF was located preferentially in the chloroplast, but also we verified this protein in the parenchyma of the xylem vessels. And in relation to the fruit traits, we did not observe, even though you can see a small size here, but it was the first harvest. But when analyzed the main anatomic and morphologic characteristics between the fruits, we didn't observe a difference between transgenic or wild type. Even when we analyze the weight or the sugar content, it's very similar, it's quite the same. And then we analyzed the seeds. We observed that the seeds germination was no difference between wild type or RPFF. And then what was nice, because we analyzed the seedlings from these seeds, and we observed that the transgene is stable because it is also detected in the progeny. So that means that the gene can be transferred to the mother plant to the progeny, and so proving the stability of this transgene in the citrus plants. And in relation to the disease, we did not observe CVC or citrus canker yet in the field condition, and we are not allowed to do in artificial inoculation field condition. We have to wait natural transmission to analyze these plants. And we did observe CVC or citrus canker yet, but there are another diseases like greening. And curiously, some events, some lines are shown more resistant. This data is very preliminary, but some lines from RPFF transgenic plants are showed good response for other diseases like greening, but it's very preliminary. And talking with Steve Lindahl about that, we raised the hypothesis that probably DSF could be trigger immune response in plant. But it's very preliminary, we're still studying this. And if so, it shows that if the plant these results can potentialize the effect of this plant, that not only confuse the pathogen that use DSF as molecule, but also could maybe have a broad spectrum resistance to other disease because DSF could activate plant immunity response. So this is in the beginning. So the ongoing project regarding RPFF involves this question, and also we are investigating if DSF could be translocated to the transgenic rootstock. We already have a single commercial variety transformed with RPFF, and the non-transgenic scion was already grafting on these transgenic plants. This work are being done by this student and postdoc, and also the insect behavior on these transgenic plants are part of the project that are ongoing now in our lab. And so, back to the interaction, okay, we showed that Zalala fastidiosa, through the current sensing, regulated the expression of genes and protein important to host colonization. On the other hand, the host can also recognize molecules from Xylella and trigger immune response to impair the Xylella colonization through the vessels. And so now we are going to focus on this interaction, but focus on the resistant plant, the genetic mechanism involved in the host resistance to impair Xylella fastidiosa. Okay, so now the focus is the host resistance. And in relation to citrus, we know that even though all the sweet orange varieties are susceptible to Xylella, Citrus reticulata or mandarin are resistant. While in sweet orange, the bacteria systemically colonize and decrease colonization in sweet orange, the opposite behavior is observed in mandarin. And so we have been working for a long time to understand the genes and genetic mechanisms involved in this time course that, in the case of mandarin, eliminated the bacteria in the plant. And so I'm not going to explain you gene by gene, but taken together, we built this model that I invite you to read this review that we published in 2020 in Tropical Plant Pathology, where we used all the knowledge that we got from this study involving mandarin, and we built this hypothetical model to try to explain this resistance. And so, very fastly, we are going to focus on two genes. This one is a receptor, a PRR receptor, pathogen-related receptor. We believe that this receptor can recognize molecules from Xylella that we named PAMPs, or even DAMPs, because Xylella can degrade its cell wall. And this degradation could be probably recognized by this receptor and trigger immune response. So we found two receptors upregulated in citrus. We are working on one of these receptors because in preliminary results, we verified that it seems to recognize some pump from Xylella, but this result is very preliminary, and I'm not going to focus on this now. But I would like to point out is that in olive, in the tolerant variety of olive to Xylella, they also found receptors like that upregulated that could be involved with this recognition of Xylella and trigger immune response. So we believe that this is very important to be studied. And this trigger response could be associated with the cell wall modification. And we believe that with the transcriptional factor that mediated the cell wall modification could be this gene REP2.2. And I'm going to focus more information regarding this. Sorry, I'd like to also to mention that we transfer one gene, this receptor, from Arabidopsis that recognize this receptor is present only in Brassicacia. In that case, is more studied in Arabidopsis, that is EFR. And this receptor can recognize EF2 from Xylella and Xanthomonas too, because as Alexandra said, they are not close related, but they are very similar, these bacteria. After I'd like to talk with Alessandra about that, because we have been working with both bacteria because the similarity between them, and we believe that if you transfer one resistant to citrus, maybe you also can have resistance to Xanthomonas. We proved that using this receptor from Arabidopsis, because this receptor could recognize EF2 peptide from Xylella and from Xanthomonas and trigger immune response. So it was published last year in Plant Biotechnology Journal and has the collaboration of Dr. Cyril Ziepfeld. And it's very nice. Please look there because the detail of this work is very interesting. But going back to this one, REP2.2, that I'd like to focus on this because the similarity with olive 2. And so REP2.2 is involved with the cell wall modification. We found this gene upregulated in mandarin. And curiously, this gene in Arabidopsis play an important role in plant-resistant to botrytis and ethylene response, since it is an ethylene response transcriptional factor. And so it raised the question for us in the beginning, that's nice, this gene is involved with cell wall modification and also with the resistant to botrytis in Arabidopsis. And then we raised the question, if could cell wall modification be a genetic mechanism to impair Xylella fastidiosa colonization? So we had indeed some evidence in papers that was published in Citrus, because this one in Citrus reticulata that is resistant, the conclusion was that perception of the bacteria involves cell wall modification and activates hormone pathway. In that case, probably, ethylene, because the cell wall. And also auxin we found upregulating this one, but I'd like to point out this cell wall modification. And in 2015, we used an analysis of the colonization of the bacteria in a susceptible variety compared with the resistant ones, and also the hybrids that show different behavior in relation to Xylella colonization. hybrid that was more resistant and hybrid that was more susceptible. And we observed was that in the resistant species, the bacteria was not able to move from the primary vessels to the other xylem vessels, to the secondary vessels, and then have a systemic colonization. On the other hand, in the susceptible variety, we observed a different behavior. The bacteria was able to move through the xylem vessels and systemically colonize the bacteria. And also, this work showed that only in the resistant variety we observed an induction of lignification in presence of the bacteria, but not in the susceptible one. So when we start work, read the papers that was published in Olive, compared Latino, that is tolerant to Xylella, not resistant, tolerant. Xylella is there, but the colonization is not so fast or so higher, and they also show less symptoms. Compared with Selena, that is very susceptible, the authors showed that some results and conclusion from some papers show that in this one, in 2016, that Xylella is perceived by the plants in both cultivars, susceptible and resistant, in which trigger differential responses strongly involved the cell wall, but both express genes associated with cell wall, but in a different way. And so the other one, 2018, showed that it was very curiously because when the authors inoculated Xylella in susceptible and in resistant, intolerant variety, they observed that only in Latino, that is the tolerant, occur in accumulation of lignification together with the genes related with lignification. So they conclude that results suggest a critical role for lignin in Xylella fastidiosa tolerance to Latino. And in agreements, in additional, and also in agreements, the last work I showed, in this work, they showed that only in Latino, when they inoculated Xylella, they observed a thickness in the cell wall only in Latino in presence of Xylella. They increased the cell wall and lignification. In relation to Selena, this morphology, this phenotype was not observed. So using all this information together, we raised two questions. Since Rep 2.2 was induced in mandarin, which is resistant, and this transcription factor is associated with botrytis resistance in Arabidopsis and secondary cell wall formation, could this be a key gene to confer resistance to Xylella? And if so, could this resistance be due to cell wall modification? So to answer the first question, we transform citrus sinensis, but first we also transform Arabidopsis as a model plant, and we got results from the Arabidopsis first, and so we transform citrus because the promising results observed in Arabidopsis. And then we transform citrus, we got 6 transgenic lines, and we choose 3 lines showing different level of gene expression to propagate and start the challenge with Xylella and symptoms analysis. So what we verified was that the clones that was inoculated with Xylella showed much less symptoms as much as the gene was expressed in the transgenic line. So here the transgenic line with different expression level of Rep 2.2 showed a negative correlation with CVC severity. It means more gene expression, less symptoms. The last one that was less expression of the gene showed more symptoms and these intermediated symptoms. So we continue work on this line because it was the best one, and also it did not show any difference when they compared with the wild type in relation to the phenotype, but was more resistant. So this result was also published in 2020 in Molecular Plant Microbe Interaction. I also invite you to see the details of this work, and the plants are now ready to go to the field to analyze the agronomic traits. And then we answer the first question. Yes, Rep 2.2 confer resistance to Zalala, but could this resistance be due to cell wall modification? So we did some macroscopy analysis using the transgenic compared with the wild type, and you can see more lignification due this red intensity color compared with the wild type. And so when you use electronic scanning macroscopy, we also observed a vessel, a thickener, the cell wall of transgenic plants are significantly thickener than the wild type. And just to remind you, these results are quite similar what observed in Latino in presence of Xylella. So now our next question is, which genes are regulated by Rep 2.2 in transgenic plants associated with cell wall modification? This is the ongoing project. We don't have all the results yet, but we already observed in the PCA that the data set are very distinct between wild type and transgenic plants, and analyze the first genes, that the first data set of these genes, we could verify a more expression of ethylene signaling and upregulated of genes involved with the cell wall modification and also with the phenylalanine pathway, the secondary metabolism. And as expected, the genes associated with lignin. And then we found genes associated with resistance to the pathogen that could explain this resistance, conferred by Rep 2.2, and also cell wall modification. Now we are doing gene expression analysis and validation and insect interaction and transmission with this gene. And as the results support that the perception of Xylella in the tolerant or resistant cultivar may trigger to an immune defense response that involves cell wall changes as resistant response mechanism. And Rep 2.2 seems to be a key gene involved in this process. So I'd like to thank you very much to my collaborators, Steve Lindau, that have been working with us in the RPFF approach, Cyril Zipfeld, that worked with us in receptor approach, Miley Melotto, that has collaborated with us when we use Arabidopsis as a model plant, and Dr. João Lopes in relation, helping us with the insect interaction. So I'd like also to thank all the citrus bacterial disease team from the Citrus Research Center, the Agronômico Institute, researcher, postdoc, undergraduate, graduate students, and also the institution and financial support that have been supporting and helping and collaborating with our work. So thank you very much for your attention.
Very good. Excellent presentation, Alessandra. It's very important. Now all this knowledge about the interactions between the Xylella and the host plants, the mechanism of resistance and pathogenicity. This is all the three talks give us a very important background to understand the other sessions of the workshop, in which we are going to address the transmission that depends on the colonization, and also on management and so on. So now we are going to move to the debate, bringing some questions from the audience. I will invite now the other speakers to join the board, Rodrigo Almeida and also Alexandra. Very good. It's nice to have everybody on board. I will start with a question that came to Rodrigo from Felix Moran. Rodrigo, considering the high host range of Xylella fastidiosa, the non-specificity of the different strains of Xylella fastidiosa with insect vectors, and the dynamic genome of this bacterium, what's your opinion about the evolutive origin of this pathogen? Was Xylella fastidiosa a symbiotic bacteria, of plants or insects which evolved into a pathogen? So this is the question proposed by Felix.
So I think that Alexandra showed some data on this. I think work by by other groups has clearly shown that Zalala is a clade that's from Xanthomonas, right? So it's evolved within the Xanthomonas. It gained the ability of being transmitted by insects, and its genome reduced at the same time, which is a common thing that happens when you start being insect associated for a symbiotic bacteria. Alexandra, do you have anything else to add to that?
Yeah, no, I know the questions were done in order, so hopefully you got some answers from that talk. But I think the question is probably right. They said, do you think it was a symbiotic bacteria that evolved into a pathogen? I think probably yes. I think we see a lot more pathogenicity in agricultural systems, a lot more evidence of disease than probably there used to be in native systems with more plant diversity. Oh, I think you're muted, Joao.
Oh, thanks. Yes. So, Alexandra, since you are on the stage, maybe we can bring another question for you from Elvésio de la Coleta. How do you intend to address questions about Zalala fastidiosa diversity and our evolution in the natural environment?
Well, I don't know if I'll be able to do it personally, but I know that there are several projects that are going on right now in at least 2 of the native subspecies ranges. One starting out in the eastern United States, where there's going to be a lot of sampling in natural environments, and another one in Costa Rica. So I think even though it's very difficult to sample outside of agricultural systems, because often it's harder to know which plants will test positive, so you have to potentially sample a lot more with a lower success rate. I think these things are really important. We've also had some success sampling in botanical gardens, so that increases the number of hosts that we can understand. But in California, that doesn't really increase our diversity of bacterial species because it's still strains from a recent introduction. So I think focusing on native ranges and increasing sampling, especially the native host plants, is the best way.
Yeah, certainly we need to study with more efforts these natural environments. There is very little information, not only in the bacteria, but also on the vectors that could bring these bacterial strains to crops. And in some cases, we have new disease originating like this. Rodrigo, maybe Alexandra too. There is a question: do you think Xylella could become a big problem for a new crop, for other new crop, just like it is today for grapevines, citrus, and olives? Do you have other new victims of Xylella in the near future or in the distant future? This is for Alexandra and Rodrigo. You can.
I'll take it. I think that I mean the short answer is yes, right? But I think it's there's a little bit of chance, right? You can think of the COVID pandemic. Why that strain took off when others did not. You can also think of monkeypox now expanding. I think so. Examples of relatively new diseases. So you have blueberry in the United States. It's not new, but it's a problem. Elvésio in Brazil started looking at olive trees, and he finds Xylella in olives causing disease. So it's kind of new in some ways. And then in Europe, for example, I mean, they have the olive disease, but you can also have old diseases in new locations. So in Israel, for example, we have Zella in causing problems in crops that are also susceptible in California. So the short answer is yes, but it's really hard to predict, or potentially impossible to predict. And I think that the example of olives in Italy being transmitted, Zalala being transmitted by Spittlebugs, is one of those that we would not have predicted based on knowledge we had. So I think that the answer is yes, but I think that there's a lot of ecological and biological complexity. So predicting these things, it will continue to be very difficult.
Yeah. Nobody could predict that we would have such a devastating epidemics in southern Italy. It's very difficult. One interesting thing that I listened from people there is that the outbreaks initiated in touristic areas, according around the Mediterranean region. Is that a pattern for Europe, as you see? But do you think this is the case for other areas?
Well, I mean, it's hard to know, right? But I think that what data has shown from Europe is that when they were surveying shipments of plant material coming from Central America, for example, they found Xylella in, for example, coffee plants that were introduced. When they started going to markets in France, Italy, there are reports from both places that open markets, they sampled coffee plants and they had Xylella. So this essentially shows that you have a lot of introductions today, particularly through coffee, because that's what they've sampled. But coffee, of course, there is no coffee in Europe. They like drinking it, but there is no coffee in Europe. It's all for ornamental purposes, right? But you go back in time and there is the hypothesis for almonds in Majorca, and the introduction in Spain there is because of introduction of contaminated plant material, almond material. And until a few decades ago, you could easily import plant material. Trade, right, was very easy to be done. Was not very regulated. So you have to assume that you had a lot of introductions. Most of them never led to anything, and a few established in Europe, in particular in Europe. The real question to me is not why you have so many introductions that have been identified. It's actually why do you have so few that have established, which I think is a more interesting question than the alternative. So today, I think it's mostly through trade of non-crop hosts or ornamentals. In this case, coffee is an ornamental. But in the past, I think a lot of these introductions were all because of contaminated plant material that people were importing for the purpose of planting for commercial purposes.
Yeah, certainly. Okay, well, let's switch a little bit of subject. We have a question here for Alessandra Souza. Alessandra, how does the insertion of the Xylella fastidiosa RPFF gene into the transgenic plant cause this pathogen confusion, reduce the severity of symptoms?
Okay, so I try to explain this during my presentation, but I recognize that it's very fast, and who is not familiarized with this system, maybe it's a little bit confused. So I'm going to try to explain it. This gene in Xylella or in bacteria is associated with this DSF synthesis, and the DSF is a fat acid. All right, so you use this gene from the bacteria to transform plants And we expect that the plant could translate this protein or this enzyme and also produce the DSF similar with the DSF from the plant. And to prove that, there are many papers already showed that indeed the plant was able to produce this DSF similar to the plant, the bacteria, which is different to the plant. It was shown by Steve Lindau's group team, and we used the same approach. So when the plant produces constitutively the DSF similar, the DSF from the bacteria, when the bacteria is inoculated in the plant, the bacteria can sense, recognize this molecule as its own DSF molecule, but not from the plant. They sense this like the all DSF from the bacteria that bacteria produce. And for the reason, the DSF regulated genes from the bacteria. And this regulation is less movement. So the bacteria express less genes associated with movement. Consequently, the bacteria do not colonize efficiently the host. That's why we observe less symptoms. When I showed here that we developed an antibody against this RPFF from the bacteria is to prove that the plant is able to translate indeed the gene from the bacteria to the plant. It was very hard to develop this antibody. Why? Because the plant has all the apparatus, genes, and enzyme to produce fatty acid from the plant. The plant also produces fatty acid. But the protein, and it has a lot of the synthesis gene that is similar to RPFF. And to get an antibody specific to the bacteria was a challenge. And so we have to analyze whole the genome from the plant to find one piece, a peptide specific for the bacteria. And then we got a good antibody now. And so we can prove that the plant is able to translate the protein, and also the team from the Steve Lindau was able to prove that the DSF molecule has been produced. And so this molecule is recognized by the bacteria and down-regulated genes involved with colonization. That's why you have less symptoms. I hope I could explain better now.
Certainly, much more clear. Although the talk was very clear, but
it's a lot of things involved.
Yeah. A lot of things. It's amazing mechanism that the bacterium develop in these interactions with the plants. And there was another question to you. Have you done any transgenic work with the RPFF gene in olive trees? If not, do you think it's possible to use this technique for that crop?
Some different question. I mean, the first one is if I have done it in olive, no, yeah, because we are not working on transgenic plants in olive. But I know that the team from Pasquale in the body, Pasquale Saudanelli. Pasquale is work. Yeah, I never know exactly his last name. Pasquale, he's work on this, but not transgenic. He's work on some RPFF approach in a different way because they are not work on transgenic there. But I don't know if it's easy to transform olive because it's very difficult to transform perennial plants. Okay, and so I don't know if how is the possibility to transform olive, but we are not working on this, only citrus.
Okay.
But I believe that could work.
Give it a try.
Yeah.
I wonder whether you guys would like to make a question to each other, or do you have any related to the other talks? No?
Yes.
Okay. Go ahead, Alessandra.
Yeah, as I told in my presentation, I'd like to ask Alexandra, how should I mention this closely related relation between Xylella and Xanthomonas, because I am one of them that in my papers I use it. Xylella and Xanthomonas are close related. And now, because your talk, I'd like to put it in the correct way, the right way. Please let me know how should I write now.
Well, as I said before, honestly, I think it makes it harder for the Xanthomonas people than the Xylella people. I'm happy to email you the reference to the paper that I cited in the talk. But it's just that Xylella is within the Xanthomonas genome, the genus of Xanthomonas. So, that it's enclosed on either side. It's pretty. I think it's pretty hard to contest that they technically are the same genus. But I don't. Yeah, I think that you could say that Xylella is a lineage of Xanthomonas. I think that would be pretty fair, if you want. But I don't think anyone's going to get too upset if you say that it's closely related to Xanthomonas either. I don't know if anyone is like. Although I talked about taxonomy, I not really pro taxonomy generally. I think it's better just to look at the relationships and describe them as they are. And if you try to name everything all the time, you just have a big list of names and not much else.
Okay, thank you.
Sorry if that wasn't very helpful, but
good. Any other questions you would like to make? Maybe no. Okay, we have so maybe we could move to some questions related to management. There was a question here concerning management of Xylella in North America. Rodrigo, could you talk a little bit more about the different strategies used for PD versus disease management between Northern and Southern California?
Sure. So the main difference is that in Northern California, you have a native vector that is under good sort of biological control, that is common but not very abundant, and that's Rhaphocephala atropunctata, or the blue-green sharpshooter. So you have some spread, but you don't have a lot of spread most years. And again, the vector is already under good control, natural control, because it's native here. In Southern California, most spread that we're concerned about is driven by an invasive species, Homolodisca vitripennis, the glassy winged sharpshooter, and that instead can reach very, very large populations. People have counted thousands in one citrus tree. You can certainly find, or you could in the past find over 100 insects on one grapevine. So the strategies are very different. So in one case, for example, vector control is not going to be very helpful. It may help, but not very much. That's in Northern California. In Southern California, for example, you need to suppress vector populations because these invasive species can reach very high numbers. In both cases, the removal of infected plants is important. But the other thing is that in Northern California, there seems to be more of a seasonality for infection. Most infections that remain chronic from year after year, or the important infections, they tend to, we think, occur mostly in spring, while some data available has shown that in Southern California, you can have infections later in the year, in the summer, for example, and a lot of those will survive into the next year. So there are some ecological differences that are driven mostly by the insect vector, a little bit by temperature as well. and those will change how management has to be done. In this particular case, again, suppressing vector populations is certainly the major difference, but not the only one. So yeah, I think that that's kind of a summary of it.
Yes, see that same pathogen, same crop, in regions that are not so far from each other, you have differences in management. And to other crops, to other regions, you have differences. So, for example, in Europe, people are having a hard time trying to stop Xylella in olives. Even with our knowledge today about several management strategies, what do you think is the biggest difficulty there?
I haven't been there for almost 10 years now. So I don't know exactly, I don't know well what's going on the ground in Puglia in particular. I think it's been a combination of, it has been hard to establish management practices because of societal resistance, particularly in terms of cutting down trees in buffer zones, as well as spraying pesticides to suppress vector populations and other strategies as well. So that has allowed for the pathogen to move a little bit more than it would otherwise. I think that there is also a lot of, my understanding is that management practices such as tilling to suppress vectors are not widely used. So you're not suppressing vector populations. And again, it's removal of plant material, infected plants seems to still be somewhat controversial. So if you're not uniformly suppressing vectors and you're not uniformly removing sources of inoculum, it makes control hard. And again, this is, again, as I mentioned, controversial, so it's more of an ecological answer to the question. I think that, yeah, if you don't get rid of the pathogen, you don't get rid of the vectors. It's hard to control Zella in particular.
Yeah, it's hard to put all the strategies in practice due to several reasons, economic, social aspects also. So it's complex. Okay, I think we are getting to the end of our session, our time limit, which is 1 p.m. Brazil, and I would like to acknowledge all the speakers for the excellent talks. The audience had here some of the best scientists talking about Xylella, and this was very important for this workshop. I think it's all the information presented here is basic for the other sessions, especially for those who are starting to work with Xylella now. So next session on next Thursday, we will talk about the transmission of these pathogens, vector transmission especially, talking about vector ecological aspects and biological control. So I advise you to look at the program to see the talks. We should be presenting, also should send a link to all the registered participants for the YouTube link for the next session. So you should receive by email. And for those who are not registered, you can click on the website of the workshop, and you can get all the program, all the information, and also these links will be available there as well. By doing so, we can finish this this session. I acknowledge all the the audience for following us, and hope to see you next week. And have a nice week to everyone. Thank you for all the supporting people too. Bye.